Hello my friends, Roger once again, Mudfossil University. What you are looking at right there is a mud fossil lung. Not only is that a mud fossil lung, it's been DNA tested and certified. This is what is now called interstitium, and there is another thing that it's called. That right there is what is known as chromatitite, and that is found in layered intrusions. Let's look at what they mean. Okay, that's a layered intrusion, and they claim it's because of dark volcanic and light volcanic and dark volcanic for some reason, which is the only way they can explain it. Let me put it that way. Now let's look at what it says. Now, it's these layered intrusions is a large sill-like body igneous rock, which is really the flesh of giants, exhibits vertical layering of differences in composition and texture. It's the muscle, it's the connective tissue, and then it's a layer called interstitium, which I just showed you, and here it is right here, chromatitite. All right, that's the layers found in layered intrusions. That is what I just showed you in the mud fossil lung that has the same coating. They separate organs. That's why they find them in these layered intrusions. Not only that, not only is it found in layered intrusions as layers, the layered intrusions aren't always flat. Sometimes they're like this. Have them explain to me how a volcano made that. These are the flesh of giant creatures. That's what layered intrusions are. The chromatitite, well, this is a close-up of the chromatitite, and it's a lung. This was DNA certified. I'm going to show you the report. That's another lung where all of this, really it's called pleura. You call it interstitium, pleura. It's fascia, it's coating. It's a thick fibrous coating that keeps organs separated from other organs. This now is gone off of this one, and we're down to the actual lobe area where you're down into the lung tissue itself. This is where you have the pleura, the coating, the wrapping, the tunica, whatever you want to call that. Now, let's look at some more pictures of lungs here. So this right here I made myself as mud fossils. This is a mud fossil. It took about 6 months, maybe almost a year, somewhere in that area, of the same chemistry, the same process that it takes to make mud fossils. Now, it's not solid, but it did set up, and I still have it, and it's pretty hard now. Now, that is the tendinous fibers. Those don't change like they do in the mud fossils. It's calcium carbonate, CaCO3 primarily, and it turns into limestone. Totally different. Well, not totally different. Well, yeah, it is totally different. All right, now we go into this is the lung tissue that it gets exposed for a long period of time. They have chunks in them, and then the blood runs through it. And this is that's the fascia where it's hydrated and it's a little more fresh. Actually, that's pretty fresh fascia. That's the same stuff as was that chromatitite. And this one here that's the lung that we had sent off for DNA certification. The blood from this lung. And that is the blood that actually just came right out of these. You see all these little blood or little alveoli, not alveoli, I forget what you call them, but they're the lung tissue, little pockets in your lungs. And then the blood just came right out of here when it was drying out. And this is what they call a smectite. It's not a smectite. This is an artery. And that artery, this is an artery right around it. And inside, well, actually, this is a vein. I'm sorry. You see that? That's the vein clamp. The vein valve is right there. That's a valve. And that only lets blood go one direction. When it goes back the wrong way, it pinches that valve closed. This comes right through here, and when it goes the wrong way, it snaps itself closed. This is a vein. The arteries don't have valves in the center of them. The arteries are wide open. So that's the difference. They think they're smectites, and they grew somehow. I don't know why they consider that to be a smectite, but again, they can't. If you can't understand something, you don't know what to say about it. They come up with things that are just ways to explain something. And now I think they have to take another look at it. That's all. Mudfossils.com or Mudfossil University up on the web, and this is the link to the DNA report for that lung. Now there was 3 separate samples sent. It was 36-inch tip, the lung, and a mud tip, and they all came back, and it was ancient protocol DNA, and they all came back as human mitochondrial DNA in 2 regions, which is all they tested for to prove it, and it was dense DNA, and here's it was. Here's the lung. They all came back for the same result, but the lung came back Homo sapien mitochondrial cytochrome B gene, and then also Homo sapien mitochondrial D loop region. So, and they were dense, and it was about 100 base pairs, he said. And these are all the CTAGs and all that. It's a certified lab, and it's a certified test. So there's no reason to say that this is not valid and shouldn't be looked at. All 3 of them were valid. Now, I showed you the sample. Let's take a look at some more how that played out as far as the samples go. So once again, you see that this is the chromatitite, which is, they find these in layered intrusions because they are fascia. This is a close-up of the bloody tissue invested area of the lung where we harvested the DNA, a little pinhole somewhere we put down in there to take out DNA inside of arterial area, which is the red area. It's soft, and it's almost like, I mean, it's coming out almost like blood. Alright, there's the DNA tested lung. And this is the blood. This is literally blood. Blobs of blood ran out, not from this one, but from this one over here. You see all those little circles in there? You see it? See all the little blobs of blood? See all these little circles here? You see the way these things make these little sculpted areas? You see this? You show this to a guy that's done autopsies, he'll know what this is. That's the pattern of the lobes of the lung inside. I'm still looking for someone in the mainstream community to confront this and maybe bring it forward to have it discussed. Because if I'm right, and there seems to be a lot of supporting evidence. I mean, I have DNA tests, I have CAT scans, I have the specimens, and I have anatomists, I have all the chemistry, I have everything. So I think it's time to do this. So someone should come forward, and that person is going to be sooner or later seen as a hero. They may be laughed at for a very, very short time. But if mainstream does not come around to this, they just continue to teach what is not correct and ignore what is correct. I see that as a very, very serious failure on their part. I'm going to leave it at that. But I mean, I would want my money back. I got to be honest with you. And I'm just going to stop it right there, because until they start paying attention, I mean, I wouldn't be happy if I knew that they didn't care enough to pay attention. You know, I don't go to them for my information, but if I did, I would want to be thinking I was paying for somebody that took the time to be sure they were saying what was right, didn't ignore facts, DNA, and all that stuff, which is commonly accepted in a court of law. And a court of law would be a remedy for all of this, because then I think they would say it's time to give the money back. But I don't know. I don't know any of that part of it. All I know is what I'm saying is factual and correct. Mudfossil University, have a sterling afternoon.